Have you ever wondered how we compare two DNA sequences to see if they are similar? In this video, I'll show you a simple visual method called a dot plot matrix that makes sequence comparison easy to understand. So, a dot plot is simply a grid that helps us to compare two sequences letter by letter, which allows us to look for regions of similarities. Usually, one sequence goes across the top while the other goes by the side. And whenever the letters match, we place a dot. So you can think of it like a crossword puzzle. When letters line up, we mark them. Let's use real examples. So here we have our two sequences, sequence 1 and sequence 2. Don't worry about memorizing them. We build everything step by step. So the first thing to do is to set up your grid. Then write the second sequence across the top of the grid with the first sequence going down the left side. Each box now represents a comparison between the two DNA letters. Then the next thing to do is to compare the letters. So if the letter in the row is the same as the letter in the column, we place a dot. If they don't match, we leave the box empty. You don't need to calculate anything, just match the letters. After comparing all the letters, we end up with this completed dot plot. And at first, it might look random, but the pattern is what matters. So usually in dot plots, we don't focus on single dots. What we look for are diagonal lines of dots. And diagonal lines mean the letters are matching in the same order in both sequences. The longer the diagonal, the stronger the similarity. When we read along the diagonal, we get the sequence CATCG. And we can also see the diagonal twice, meaning the first sequence appears twice in the second DNA sequence. In recap, dot plots help us visually see similarities between sequences. To do this, we place one sequence across the top of a grid and the other sequence down the side. Whenever the letter in the row matches the letter in the column, we place a dot in that box. When these dots line up diagonally, it tells us the sequences share the same letters in the same order, and the longer the diagonal line, the stronger the similarity between the sequences.